last week we sort of stopped, and at the point where I was just discussing about how to code things with neurons and how neurons, at least in biological systems, encode the sensory world. And I was talking about a cat, and I'll just draw that again. Little cat here, looking at a screen, and on this screen you put some sort of basic sensory thing, like for example might be in different directions, and this is the stimulus we're encoded through some sort of brain activity. And what we do is we put an electrode, a little piece of conducting material, in here, and we record the activity over time, the electrical activity, look, like this in over the course of, let's say, maybe that would be 20 to 30 milliseconds there. And you're recording this at fairly high frequencies. And then what you do is you separate this out, and you can look at different frequencies. And typically you'll do some sort of, I didn't mention this last time, I just mentioned the top one that I'm going to draw. You'll basically see some sort of high pass filter like this. And this is picking up these particular spikes. And then what you do is you hold, and then you'll count the crossings, and you'll put a line, and these lines will be spikes. And they occur because, if you recall from last week, you have, when a spike happens, inside and out of the cell, you have very rapid crossings of positive and negative ions, and that causes the space outside of the cells, the neurons, to become more negative, and inside more positive. Since this is an inverse sort of relationship, it's locally, you can think of the being, it's not a closed system, but it's... Sorry?
Yeah, I was confused about, the spike going down, but that's what you're talking about, unless there's also this restoration period afterwards, I suppose.
Yeah, exactly. So locally, the total number of ions and their net charge stays about the same. But when a spike is happening, because we're recording from outside of the cells are drawing in the positive ions, and then the net charge outside becomes negative. And these spikes will have slightly different shapes. So then we can analyze these different shapes, and let's say that these two look similar, and we'll label those as belonging to neuron A, and then this one here might be belonging to neuron B. And we do this with some statistical basis, and sometimes even by eye, actually. You just look at these waveforms and match them up. Some old electrophysiologists will do it by eye, but most modern people do some sort of statistical method. And the principle is that, well, these neurons, we have our electrode here, and these neurons are, in different locations around. And the exact way that they're orientated and so on will cause there to be a different sort of wave of electrical change that's detected at the electrode tip. And therefore, on this basis, the changes in the electrical field will look different from these different sources, and therefore we distinguish the sources based on this. Perfect, of course, because in principle, two neurons could have similar profiles, but that's the way that we do it. And we call this unit multi-unit recordings.
So in this statistical analysis, how much of the variation is explained by positioning and how much is differences in cell type or age or configuration or something?
Oh, yeah. Yeah. So that's also interesting. So, yeah, that people claim, and I sort of believe them in some ways. Broadly speaking, it's true that you can determine by the shape of these things whether or not the cell is a positive or negative cell, like an excitatory
Oh, really?
cell. Also, yeah, the geometries of the cells will look a little bit different potentially. The inhibitory cells, as it happens, typically are much smaller in size, but also much more varied in size than the pyramidal cells, which are the main excitatory cells. So there's actually quite a lot of variation, and I think it's never perfect, the sort of guesses that you're making, but they're pretty good, and I think it's at least theoretically even, and practically definitely quite rare that you would have two of these units that look identical but are actually coming from separate sources. Most of the time they look fairly different, but of course the more data you collect, then the more confident you can be about there being a difference or not. Yeah.
So how good does this recording get? I mean, people put in 100 of these things, and then you can say with like high confidence, here, there's 20 neurons, this one fired and that one fired, and we understand exactly what happened there, or is it?
Right. So yeah, so that's actually, so this is a single tip electrode, and of course, what you can actually do is you can put in multiple electrodes. These are called tetrodes, so little clusters of electrodes together, and then like different shaped things like in a cluster. So this neuron over here and this neuron over here will have some sort of delayed. You'll see the activity happening at the closest neuron, the closest electrode before the far away electrode, and likewise for this one. And then on this basis, you can more confidently distinguish the sources. And another type of electrode that's quite common are these multi-laminar ones. So they put sort of a ceramic material in between different contact points. So there'll be like multiple contact points like this. And the distances here might vary between like 5 micron and 100 micron. It really depends on what you can make these in different sizes and variations. And in total, when you do these sorts of things, and there's one probe which is very popular at the moment, which what's it called? Neuro. I've forgotten. But this can, you can with just one of these, you can easily record up to 10,000 statistically different units.
Wow.
Yeah, and there's, if you have a collection of these, you can easily get into the 100. So there's right now this deluge of data, which frankly I don't think many neuroscientists know how to grapple with properly, me included. It's really overwhelming how much data there is. So it's actually a great time to be in theory. Because of all this data. There's a little bit of like experimentalists spend a lot of time and money and stuff to collect this data, so they can be a bit protective of it. But there are open repositories of data, and one particular organization which I have used the data from is the Allen Brain Institute.
Right.
And this is I don't know if anyone else heard of them, but there's like many Allen institutes in Seattle, Allen AI Institute, Allen Cell, I don't know, different ones, and there's one called the Allen Brain Institute, and that's quite good. It has a lot of data, but it's not all electrophysiology data. So this whole thing where you are recording electrical current is called electrophysiology. electrophysiology. And another thing that you can do is since when the action potential comes, and one of the things that happens at the synapse is influx of calcium ions, you can actually put a little kind of marker in here that is going to detect when a calcium comes on, and then it will light up in a particular color. And then you can track these, the sort of the calcium movement in and out of the cell. And using this process, you record from lots of neurons. So actually, you visually, instead of putting an electrode, you actually scope and look at some little area, and you see different neurons light up, kind of like a Christmas tree.
Sorry, how do you get the
over time
the fluorescence? That's like some sort of gene editing? Or?
Yeah, the fluorescence. Yeah, it's a genetic marker that lights up, that fluoresces when calcium enters the cell.
Uh-huh.
And then you just use some optics to look directly at some tissue.
So these are in mice or something?
Mice.
Yes.
Yeah, so these, not with rats, since that technology hasn't been developed. In theory, it could be, but mice have been the primary animal to use. I think they are developing, or there was talk of marmosets, but yeah.
Yeah, this actually gets to one of the questions I was going to ask. So when you're talking about spikes, I mean, last time you described both chemical and electric sort of computational pathways. So you have these ions moving around, and also there was some chemistry. And I think I asked you which one was sort of more primitive in terms of being older. Could you briefly run through again what the trade-offs are? So the electronic form, like the spikes, is fast, and that's its advantage. Maybe both fast in terms of actually moving around, and fast also in terms of restoring the state, so you can do it again. Perhaps I'm guessing. I don't know.
Yeah.
What's the other? I mean, why not do everything electrically? Why is these other ingredients there? What's the
Well, when you do these things electrically, you're sort of relying very heavily on sort of passive mechanisms, and the sort of, like, for example, the passive just geometric structure of the space that the electrical current flows through, or that these ions diffuse in, and so you sort of have less control over this. I mean, you I suppose insofar as you, let's say you have less active control, I guess. So it can't be as easily modulated. If you have discrete synapses, like chemical ones, you can modulate that with some. You can block the receptors, or you can rate limit the process by saying that only a certain number of these receptors exist, and they can't all be occupied, more than once per 30 seconds or 10 seconds, or whatever it might be. So this kind of, and then you have like ion concentrations that need to be reset, and lots of different mechanisms which basically let you slow down or modulate things. I think probably that's the main advantage of not doing everything electrically or passively. Yeah.
Is there any role for magnetism in the brain? I presume not, but I mean, it's striking that in our early computer systems, memory was done for a long time magnetically, but in the beginning using these flux wires and stuff like that.
Right. Yeah. Don't quote me on it, but I do believe that migratory birds have some evidence to say that they can pick up the magnetic field of the Earth.
Is that actually
in order to navigate,
or is that some other organ?
I don't know, actually. And I might be completely.
But anyway, it's plugged into their nervous system somehow. Yeah.
Yeah. But yeah, I mean, so the other thing that I was going to mention, which is kind of, I suppose, ends up being related, is that kind of related to this picture in the top left, how you see these electrical fields kind of coalescing and interacting and so on. To get the spikes, you do basically a high pass filter. And then you can also do a low pass filter. And then you get something called the local field potential. And this has, lots of different features depending on how you filter it. Local field potential. And this is a slightly controversial signal in the sense that people have found through a lot of different sort of correlation studies that when the band that of 20, 30 hertz or whatever it might be, typically we're talking about something like 5 up to 120 hertz, something in this range. They'll say something in this range has a high power during this moment, and then over in this area of the brain it causes or we observe a correlation that this set of neurons fires with more preference, or more easily, something like this. So there are lots of these sort of correlation studies which basically this signal, the local field potential, or LFP, is maybe sending kind of long range, low frequency information across areas. It is also true, though, just biophysically, that when different parts of the brain, in various parts of the brain, the primary cells or the pyramidal cells will kind of line up. These are the major excitatory neurons, and then this causes, if they all fire and they're all directed in the same way, then this can cause a very dramatic electrical field across this space.
Antennas in your brain, then.
Right. And it's actually, this is, these things that happen are actually the basis of, for example, a technique called MEG, where M is for magnet, something about magnets, magnoencephalography. Yeah, that's right. So this is actually the, what lets you measure this stuff. fMRI is a bit different. That one, you're actually measuring the deoxygenated,
right,
or oxygenated red blood cells. So this is for fMRI, because in fMRI you get there's a correlation called the blood. Let's go to another slide, actually. You get something called the bold response, which is the blood oxygen level dependent response. And so in this case, you're actually looking at, you've got your neural matter here, and you've got some blood vessel. Maybe let's do that in red. And this is flowing through, and when the neural activities are stronger, then you have more exchange of, or you have more deoxygenated blood. It uses oxygen to do the neural stuff, and then that makes a difference in the magnetism of the blood, and then you measure this difference over time.
So that inverse problem is so complex that if you can solve it, why not also solve it for, I don't know what's part of the spectrum these signals are in, that are these global signals you're talking about, but how much of a role is that I mean, you described it as though it was like some low information signal that other parts of the brain could then latch onto and do something, but it's not like you're sending, megabytes of data per second. See you, Tom.
Was he writing something on the board? Because I cannot see anything here.
No, I can't see anything either. Maybe it timed out. He'll be back. Maybe he'll be back.
Yeah, maybe. Oh,
There he is. Hi, Tom.
hey.
I'm back.
All good?
Alright.
Yeah. Did you missed?
Oh, I was starting to ask a question, but it's no big deal.
Okay.
I can repeat. Yeah, I think you were writing something or trying to write something, and we didn't see anything on the second board here.
Yeah. Okay. Yeah, so I was talking about the bold response. Can
Yep.
you see bold now?
Mm-hmm. Yep.
Okay.
Yeah.
This is the blood, oxygen level, dependence, response. And the principle of this is that you'll have some sort of neural tissue, and running through this tissue, like all tissues that are healthy and operating, you'll have blood vessels, and you oxygenated blood to perform different functions, and then the deoxygenated blood actually has a different magnetism. So you are looking at the magnetic effect of the red blood cells being in their oxygenated or deoxygenated forms, and it's premised on this interaction or this response that happens.
So there's a presumed correlation between the neurons doing something and them consuming more blood or more oxygen.
More oxygen. Yeah.
Mm-hmm.
But for a wide variety of reasons, there are different people have a sort of critical opinion of this procedure, and it's widely used it is used in fMRI studies.
Yeah, you mentioned the salmon.
Yeah. So for anyone that's missed that, you can just look up fMRI and a dead salmon, where they found, that this dead salmon was responding significantly to different things. And I recall that when they added some extra statistical complexity or sophistication, namely by basically lowering the chance that you detect a false positive by essentially lowering the group, like or the sort of the threshold by which you declare something to be statistically valid according to the number of things you're measuring. This correction is called what is that called? Multiple comparisons correction.
Mhm.
Yeah. And so in statistics, you sometimes have like some alpha, which you set to be in classically is 0.05, and then you do some statistical test, and then you measure, is it below or above this critical value? But then what you do in the multiple comparisons is that you multiply that by, the number of comparisons you make or something like that to make it smaller. Or sometimes you do some other. It's not necessarily very theoretically. There's not necessarily like a rule about how to do this. It's just different systems or conventions develop. It's also a bit like this number conventionally being 0.05. If I recall, it was just some famous statistician in the whenevers some decades ago said, "Oh, you should have about 1 in 20 chance or something." So then it just became that. Yeah, but other people sometimes use 0.01, or they'll report things like, "Oh, this was a highly significant thing if it was less than that," or something like that. I don't know. It's just this is all very well. It's a bit people these days, I think, especially in the computational neuroscience community, treat different older statistical methods with a bit of disdain, perhaps rightly. And the fMRI folk are being much more sophisticated these days, and yet still criticized. I think, sometimes rightly, but it's good that they're, at least from my perspective, getting to be a bit more rigorous. But they're always going to be, unfortunately, just through the nature of things, a few steps away from the actual neural activity, just through the nature of the recording.
So if I
electrodes in people's brains for.
So the premise behind fMRI studies is something like location matters. The different parts of the brain do different things, and the more fine resolution you have on where it is, then you can make conclusions about what it is and how it's working. Is that kind of one of the principles involved here?
Yeah, classically that's what it's been about, although in the last few years there's been more and more controversy about the idea that there are specialized areas in the brain.
Right.
Because it seems that, and I mean, I think it's obviously true that there are specialized areas. They're not so specialized that, that they only do that exclusively. So there's been sort of a group of people who've made it their mission to find, let's say, a cell that responds to sound in the vision area or vice versa. And it's pretty routinely done now that you can find, or you find a cell that responds to very particular stimuli in an area of the brain that we thought was only responsible for very simple statistical features of images or something like that. So
could we briefly go through
these exceptions back in the trend?
Sorry to interrupt you.
Yeah.
It'd be useful to just very briefly go through the high level basis for why we think different areas of the brain do different things at all. I mean, are there... One thing would just be inputs are wired physically into different parts. Another would be these kinds of fMRI studies, which show different levels of activation given different modalities of input. Or, are there different kinds of neurons in different areas or different physical structures? What's the kind of basic argument?
No, the main lines of evidence would be things like fMRI in different animals, but also it's like lesion studies,
right?
Or ablations, you would say, in the machine learning world, right? And sometimes these lesions, or brain injuries, strokes as well. These wouldn't be intentionally given, but they would be noted that, people that always have strokes in their temporal lobe have problems, recognizing faces or whatever it is. And so these kind of correlation and case study kind of lines of evidence have been pretty classical, and fMRI is normally clarified and shown that most of those things seem to be broadly true. And yeah, in terms of what's the same, well, actually, so there's different areas of the brain, of course, kind of levels as well. The very top level is called the neocortex, and there are people that this is uniform almost across the whole neocortex. I don't quite believe that, but I agree that it is highly regular, and it's highly regular in that. So it's actually the very outer layer of your brain. So if this was like the beginning of your skull, then this is the midbrain here, so a bit more inside of your brain, and this might be like let's say half a centimeter to a centimeter in thickness. Then you can sort of break this up into different layers of neurons, and they have different sort of compositions of neurons, and there are classically we say about 6 of these layers. And then you might have some, and that they're sort of organized. What's interesting is that if you look from the top down, you'll see that they're kind of organized in these little columns.
Top down,
and then each column
outside in.
Yeah, so we're looking like from like down onto the top of your head, let's say.
Yeah, okay.
Or the side or something. Whereas this one, we're looking at like a cross section. So this is the. So it's like if we're looking down like this is the top, and this is inside. And then here it's like we're looking straight from the top. Yeah. And these different columns have different specialities within a brain region. So the most famous example that I know of, for example, is that if you look at a rodent, it has whiskers, and each of these whiskers will actually map to one of these columns. This particular area of the brain is called barrel cortex. I have about this area of the brain, starting from the whiskers themselves and the mechanoreception that happens there, even the physics of this thing, or mechanics, let's say, all the way up to this part of the brain going through the different stages. And in that, there's a picture of this neocortex as well. So, and what I know about, and when I was going through all the evidence, was that, there's general sort of trends that occur not only in this part of the cortex called barrel cortex, which probably has the most pronounced specialization of these columns of any brain area that I know of. There are these like general sort of trends or commonalities that happen in the different layers, and there's a general sort of scheme that we think that the information into the 4th layer goes and gets processed in the upper layers, and then goes down to the bottom layers and then gets processed. And at these top layers, you get crosstalk between the columns, and at and then at the bottom layers, you get long range interactions coming from other places, like other brain areas. And this kind of, some of these general themes actually run true for other brain areas in the neocortex, other cortical regions, we would call them. And but there are many exceptions or specializations in different regions, and ultimately, though, the output tends to be from the midbrain out to have some sort of feedback on the midbrain or long range connections.
This organization into columns you're talking about, this is developmental, so it happens during the development of the brain, or is it part of the learning process that you get this sort of emergent
it's part of the development. Yeah, mostly part of the development, but the exact kind of like mappings between, let's say, whiskers and their columns, or eye regions, and things like that. Also, in the visual cortex, you get columns which are just for color, and then others which are for black and white. This happens as well.
Sorry, I thought you were going to say, so the precise mapping, is it every mouse or whatever has, I don't know if you can compare columns, if they're that regular that you could say this is.
They're pretty regular, actually. Yeah. They're like rows and columns in this, like in the columns themselves, looking top down.
So is it like
that
every rodent
onto the side of the face?
The front left whisker on column 2, or is it somewhat random?
Something like that. Something like that. Yeah.
Oh, really?
It's pretty. So in that case, it's quite developmentally fixed. However, and I'm sure someone's done this. If you remove one of the whiskers or damage it, then that column will probably be repurposed and maybe invaded, functionally speaking, by the neighboring columns.
I see.
Yeah.
So you get smarter rats if you shave their whiskers.
Well, you get like some smarter whiskers around, like to make up for the missing whisker.
Right.
Yeah. So you do get like a whole bunch of these, like and another classic example is like, so back to cats, because they didn't get enough torture. Typically the back region of the brain is for vision, but they blinded the rats, the cats, and then they saw that these these visual regions were sort of functionally invaded by like auditory stuff or other stuff. And then in fMRI studies, if you look at someone who suffers from congenital blindness, then you find that their area is still responding to stuff. It's just not vision that's making it respond to stuff. Yeah,
interesting. Thanks for answering all my questions. I feel like I probably derailed you a fair bit from what you were planning to say.
No.
Get back on track if you wish.
It's totally fine. It's nice that I can feel like I'm semi-expert in something. So, all right. So I think I wanted to. So I mentioned something about the local field potential. Oh, okay. It's missing what I wrote here, but I'll just write LFP, local field potential, there on that first slide. So something that I guess I wanted to mention is that something about the neural coding, right? So let's start a new start thingos. Let's check. Is that OrbCam? No. Is this OrbCam still following you?
It's not following you. You can take back control. I took it when you left, so you would appear here.
Yeah. I see. Yeah. Okay, then. So that's good. All right. Okay, so I wanted to mention a little bit about neural coding, and let's start with like one of the most classical receptive fields from one of the most classical receptive field. Well, the thing that started it all and it was with cats, of course. So this is the visual space, and I'm going to draw how many of these? 1, 2. I think. Yeah. 6. So they shone light, just pure. So this is the visual field, right? It's like the TV screen. And then these are just X and Y coordinates to keep a track of the different space. And what they did was they shone a light in the center, small one, at the top, again, small, on the right, on the bottom, on the left. Oh, whoops. on the left. And then they did a really big one, like across the whole field. And maybe how should this? Yeah, I think I'll tell you what the spikes look like over time.
Mm-hmm.
Oh, so we shine the light over this part of the space, and then we observe the spikes like so each of these vertical lines are spikes, and then going this way is time. And this one we see something like this. This one we see something like this. This one we see something like this. This one we see a lot of this. This one we see nothing. Completely blank.
Mm-hmm.
And question is, if you look at this this area, this is one neuron, the activity of one neuron, and the question is, well, what's going on with this neuron? Well, what's happening is when it's in this center region, it's being inhibited. So the activity is actually somewhat constant here, but it's being quiet whenever we're in the center, like here, here, and here. So, and then on the outsides, it's being active. But if you activate the whole thing at the same time, whoops, sorry. If you activate the whole area, then it's going to overlap, and these things are going to cancel out. That's why this final one, you get nothing at all.
Mm-hmm.
So these receptive fields. So this is a type of receptive field.
So receptive field, the field refers to the part of the input space which is being responded to by this individual neuron.
Exactly. Yeah. So in this case, it's kind of like, it's a multi-dimensional. Well, it's not really multi-dimensional, but it is two-dimensional at least, right? It seems. You might say that. Well, yeah, it's restricted in space in this, like it doesn't respond above or below those two lines there, but then it also has a response which changes according to this X dimension. So it's at least two-dimensional. But it's clear that it's a little bit more complicated than like, okay, if I'm in this convex region, then I turn on, and if I'm out, I turn off. It's got this middle part that's kind of complicated, right? Or different. It's being suppressed and then activated in different parts. And so this implies that we're not looking at a cell that's directly connected to the sensory input. There must be some process here which is sort of also receiving sensory input and then suppressing this part. So this is one way. If you look at the complexity of some neural response, by just sort of mapping it to some basic stimulus, you can figure out approximately what kind of level in the computational hierarchy, just as a rough phrase, it might be. And if you look way down in the retina, which is at the back of the eye, then in the retina, you're going to have, cells which are very basic, and they're not going to have this kind of situation typically. Now, I have this kind of, like in this case, I've sort of implied that neurons respond in some kind of binary fashion, but that's not actually true. The neurons can respond and code for things using the rates of the firing. So if they spike very frequently, that can mean that they're you know, they're more like that. They like this stimulus more. They sort of do something like if the bar is orientated this way, maybe you get no activity. If it's orientated this way, like on the visual field, maybe you get a little bit. If it's like this, you get a lot. And then if it's like that, you get a little bit. And then if it's like if I gone too far, let's just go somewhere like this. I don't know, maybe only a little bit, maybe. And then maybe this one has one spike. And then when you plot the orient.
So each one of these lines is a spike. That's that.
Yeah, these are spikes here.
Mm-hmm.
And my idea was try to show there's a lot more spikes on this orientation of the bar.
Okay, so when we say that a neuron is responding to an input, what we mean is the existence of spikes, the frequency of spikes, how grouped they are.
Yes, the frequency. So like in this case, the spikes per second, let's say.
Yeah.
Mm-hmm. And so this would be like maybe 60 spikes per second, and this would be none at all, and this is the orientation, and it'll have stereotypically some kind of Gaussian shape like this. And so then this is its preferred direction or orientation. But you can replace this with any other stimulus feature, and then you say it's tuned or selective for this particular thing. And it can be sharp. It doesn't necessarily have to be super Gaussian like this, but or super spread out in the parameter of that Gaussian. But so this is called and basically all of AI models use rate coding, right? The idea is that you have a floating point number, something like, for example, between 0 and 1, and then this indicates some magnitude of how much how much you can sort of map this to this. You can say this is 0, this is 1 in a rate coding sense. But obviously, neurons have a temporal component to them. One criticism of rate coding is that, well, if I see a single spike, I don't know anything, actually. I don't know if I'm at my tuned or selective feature or not. I might be, but at that moment, just with one spike of information, I have no idea. And I need to actually observe for a certain amount of time in order to measure the frequency. And also I have to measure the frequency. I can't just like see something immediately and then say, oh, well, that means that I must have some sort of activity that corresponds to this orientation or this stimulus feature. So this is one criticism of rate coding. It's less efficient. You have to spend more energy. You have to send lots of spikes. Each spike costs quite a lot of energy. So the brain has come up with many clever ways of coding, which does not include rate coding. And if there are no questions about rate coding, I might just go around and talk about a few others around the corner.
Yeah, sounds good.
Okay.
So would you say AI has adopted a focus on rate coding just because they're ignorant bumpkins, or is rate coding actually the most important thing, or what?
It's probably the most easily studied, or the most widely studied, at least originally. But also, so like there was this. Okay, so yeah, now you've got me. I'm going.
Good.
So there was a famous. Okay, so all this cat stuff was done by two fellows named Hubel and Torsten Weisel. Actually, he was pretty instrumental in setting up my current institution, Waste, which was pretty cool. So I got to meet him. So they developed and they got the Nobel Prize in the 50s. Oh, I know they did their thing in the 50s. Who knows when they got the Nobel Prize? And they were really showing for the first time these receptive fields. And they were also, basically showing that you could measure the rates of the codes, like the neural activities, and it does correspond to, would correspond to the activities of, or the stimulus, basically. And this, this really took off, basically, and it developed into this whole thing, which people call systems neuroscience. And I typically put in brackets or think of this as like sensory, as well as like sensory systems neuroscience is more or less where it started, but people use the same premise, which is basically have a highly reductionistic stimulus and then try to find correlation between or correspondence between that stimulus and some neural activity, but almost always in the rates, because this was just easier to do.
Yeah.
Because the electrical instruments and stuff were not as sensitive. You weren't guaranteed to pick up every spike and stuff like this. So you're going to kind of pick up the loudest stuff. But then there was, so then like later on, there was this, there was sort of, and I know this more from a Japanese perspective, and others would know it from a more international perspective, but there were a few researchers in Japan, including people like Amari and Fukushima.
Yeah, the old school neural net guys.
Yeah, and so Amari was doing stuff where he was, and I mean, like people like John Hopfield were also doing similar things, and other people were doing these associative memory networks, which I've talked about before here, and that in a sense was still premised on these kind of rate codes as well. Like basically you have a vector of memory, and you basically say that the activities of the neurons are binary, and that some particular vector is one memory. And normally they would say that, well, when, like each of these elements in the vector, when it's a 1, it's basically like an on in some neuron, and when a 0, it's off. But as we've kind of learned, like that might make sense if you're thinking about this in terms of linear algebra, but from a biological perspective, this would kind of be saying that like this one is spiking consistently and this one is not spiking at all, which, in order to like, I don't know, maintain some memory item, I don't think there is some studies in working memory to say that that's maintained over short periods in order to remember something. But to remember my grandmother's maiden name or something like that, I don't, have a few neurons which are just consistently, or like a vector of neurons that's consistently firing or not firing to remember that particular thing, right? Or like when I recall that piece of information, it's not thought in modern neuroscience that this is what happens, that you somehow have a collection of neurons which all become active in particular ways, and instantly so, in order to recall some particular memory. So, and the main reason is it's inefficient, but this sort of associated memory stuff, I think, is partly what caused rate coding to be widely adopted in these systems. But then also put people like Fukushima, he had the neocognitron, which was kind of just like a very idea of a CNN, and then when, I'm just kind of drawing roughly what I remember from his CNN things. These were kind of like the different filters up here. It's not too important about the actual architecture, but the point is that this was also using rate coding. So I think people just, from the experiment rates, because they were kind of like the loudest, most persistently active neurons, and, the most reliable to record and so on. And then this just kind of, I think, caused there to be more data, and then more data meant more theorizing about that data. And then, there are probably other reasons, like, for example, from a technical standpoint, you can differentiate these kind of continuous values when it's not like a binary vector like this, for example. So there's probably way more reasons than what I've mentioned. But from a neuroscientific perspective or history of neuroscience perspective, maybe this is one reason. But let me tell you about some non-rate coding methods. I think I have to tell you one of the most exotic ones first. And this is like, how would you call this? There is a fun name for this. Polychronization.
This must be cool.
All right, so let's see if I can do a nice picture. I might try to use. Okay, I'm going to try to use the whole board for this one because it's worth it. So let's have 5 neurons. 2, 3. Label these B, C, and D. And then let's have neuron A over here. Let's have neuron E over here. Okay. And neuron B synapses to E, B, C, and D synapse to E, but they have a delay. So the delay is accumulation of, the length of their axon, the, dynamics of the, and the dendrites and all this stuff contributes to delay. So let's say from B, let's make, I think, 8 milliseconds. Actually, I might just not write. I'm not going to write milliseconds. I'm just going to write the integer 8 there, and then 5, 1, yeah, and then we'll do the same here, and let's do. I think we can just do this. So 8, 5, 1. Okay, and so let's have 3 scenarios. Scenario 1. Scenario 1 is that, so we're going to have, this is our spike like counter for A over time. And we'll have B, C, D, and E. So I'm going to draw short vertical lines through these lines, these horizontal lines, whenever, so this is C and this is B, whenever a spike arrives there. And let's start off by having B, C, and D active at the same time. So what's going to happen? Well, B is, going to send a 1 millisecond signal to here. D is going to send 1 millisecond signal to there. This one, like at 5 milliseconds.
So you mean a single spike at 5 milliseconds?
Yeah. This one here, right? And then I think this is right. And then let's say as well that in order for B or E to spike, that it needs 3 spikes to co-occur in order for it to reach its threshold to spike, right? So So that's the complexity. That's the cool complexity thing. So now let's do another case.
Prime numbers in your brain.
So let's do the order of DCB. So DCB. And what's going to happen here? Well, it's going to go like something like this, and then this will be trailing off. So the order of CD is what causes A to fire instead of E. And then probably you can guess what will happen in the third case. It's just the opposite, right? But for complete.
Let's see.
And then but this one does fire. So here this sort of highlights quite a few things. The first is you've got this idea of the delays. You've got the idea of an order of spikes, and also sort of the delays and I guess timing, right? Actors sort of working in concert, the time or the delays, as well as the exact order of these spikes, which is I suppose related to the timing or the delay. And this paper, it's an amazing paper, and the guy is Zikovic? Yeah, Zikovic. Okay. And there's a whole kind of like this paper was like a purely theoretical paper, and or computational paper, and I think there probably was some evidence about kind of related stuff, but after this paper, it sort of popularized the idea of so-called syn-fire chains. So syn-fire chains are like synchronous firing chains, so particular orders of neurons being active at particular times, and or sort of sequences of these were sort of thought to be important. And there is actually, I remember after reading this paper, I looked up, on Google Scholar, like who's most recently cited this, and I found this, and I haven't been able to find it since, but I found this amazing theoretical idea of like, okay, well, what if the brain uses like prime numbers to like delay things such that it organizes its activity around something. It was highly
the first thing that occurred to me when you wrote one was least common multiple and GCD.
So there could be something in that. I don't recall if I don't think this is caught on in the neuroscience community, but
well, it's related to there was speculation by Stephen Gould in one of his books that some prey animals actually choose prime intervals between their like major nesting seasons. Like they don't really reproduce in large numbers every year, but they choose prime numbers so that it's harder for predator species to kind of reproduce every second year, because they know that every fourth year there'll be a lot of prey or something.
Oh, okay.
So yeah,
that's brilliant.
I think it's probably unrelated to this, but it's not the only incidence of like prime numbers being an obvious kind of trick to coordinate something.
Yeah. Oh well, there you go. Experimentalists, go find us some prime numbers in the brain.
And then zeta functions, and then
Oh yeah, modular forms.
That's right. Yeah.
Yeah. Okay. All right. So that was like probably one of the coolest ones, and I'm not going to claim that the others are quite as cool, but they're still pretty cool. I suppose, relating to the same thing before, you can sort of do things using latency coding. In general, there's basically this whole idea of like temporal coding in opposition to rate coding.
And
so latency coding.
How does that distinct from what you just said with the SynFire coding? I mean, timing of the spikes matters. That's a special case of timing mattering.
That's just like a type or an instance of a way to encode things using temporal, differences. Yeah. Another sort of like clear example of latency coding showed up in, I think it was the retina or early visual system. And that's where you have kind of like, so in the rate coding example, on the y-axis, this would be like spikes per second. But here you put latency. And then here you put your like stimulus thing and like the different preferences. And then what you do is you have, for example, like one thing like this. And then let's do it in blue. One thing like, I don't know, like this or something. And then what you do is so this is like neuron 2, neuron 1. These different latencies. When you look at the neurons together, then different stimuli will cluster in different places in these two different latency, in this kind of like joint latency space between the two neurons.
Not really following what this first graph means. So what's the x-axis?
The x-axis are like different stimuli.
Oh, okay. So this was like before, like an abstract axis that might represent orientation or some other continuous parameter. And then you're saying neuron 1 is sort of tuned to whatever that stimulus is, x. But what, and the y-axis, I mean, before it meant spike rate. Now it means...
Yeah. This might be like, I think normally we would say like the latency from the stimulus input. So for example...
I see.
Yeah, so even though these two neurons are like occupied in the same kind of space, the like physical space, and they synapse onto the same postsynaptic neuron, the order or the latency of which their information is sent is different due to, I don't know, the dendrite structure, the synapse structure, whatever it is. And then by looking at the soma, and particularly the initial segment, you can sort of cluster similar inputs into different regions of space, latency space. Yeah.
So this is,
and then that means that,
So this is a function of like the physical architecture of the neuron.
Right. And then this means that you can do kind of slightly cool things, like you could shunt or further delay or modulate the delay or the latency of these different neurons or different dendrite segments or whatever, and then sort of block off access or change the coding in this space somewhere or another.
I see.
Yeah. Another thing.
Has the success of deep learning or sort of ConvNets or any of these models of particular, maybe limited domains, has their success shifted people's opinion about the relative importance of some of these different mechanisms, or was it sort of already sufficiently well established experimentally that basically it made no difference to people's judgment and we sort of know what's happening in the visual cortex or whatever in our brains, and if neural nets can do it without latency coding or spikes, then good for them, but doesn't really change our understanding of our own way of doing it?
Oh, I see what you mean. Well, yeah, I suppose the people that would use an artificial neural network to then claim something about the brain in terms of like knowledge of the biology, or saying that if we observe some structure in the artificial neural network, then that structure must also exist in the biological neural network. Since the artificial neural network uses rate coding, the people that would say this, or typically uses rate coding, the people that would say this would probably be neuroscientists that quite definitely subscribe to the so-called manifold hypothesis, which is sort of an extension of the rate coding model, where you just say that the distributed collective activity of all the neurons is encoding the stimulus or cognitive action or something like that.
Activity meaning just the summary statistic of the rate of firing, or
yeah, right. So like, neuron 1 is spiking at 0.2 times a second, and 99 is not active, and 162 is always active at this moment. And then you collect all these points, put it on a manifold, and then you say, okay, that's this point here. And when we move, when we observe this animal walking in a circle, it actually walks in a loop on this manifold of activities, this kind of thing. And then this manifold has particular geometry and topology. So I sort of partially subscribe to this, but I think that obviously rates are not all there is. And I'm trying to remember your question more precisely. It was something like, do people believe that?
Yeah, I could state it more forcefully, I guess. So why shouldn't I believe that maybe these other things are going on, latency coding and SynFire coding and whatever, there's 100 other things. But I could say, well, they may be incidental, or, they're sort of for redundancy, or they're doing something not super important. And if I somehow magically turned it all off, well, then I might be a bit slower, or a bit like making bad predictions in some limited cases, but basically my brain would keep working. How do I know that's not the case?
Okay, well, one example, a sort of famous example, but it's slightly controversial according to at least one very famous auditory neuroscientist that I know, and that is a head direction, or like sound localization using your head direction. So you have two ears, you hear a sound over here, and it reaches this ear before the other ear. And the distinction between, this temporal difference, however small it is measurable in, the order of milliseconds. And yet we can reliably determine where the sound comes from. And clearly in the order of milliseconds, you don't have time to look at the rates, right? Because this is all happening, one spike is like about 1 millisecond, and you have a few milliseconds between spikes. So you have to somehow determine the where a sound is coming from in very quick amount of time, and in order to do so, you can't use rate coding. So there are many different behaviors like this. Basically, very quick reactions are a common example where it seems impossible to use rate coding.
So sort of drive-by-wire, the physics of the sense organ just directly hook up to something.
Right. So, I mean, you probably could still work it out or do something with rate coding, but you wouldn't be able to have that same reaction speed. Yeah. I would say also maybe one kind of quirk or sort of side quest that's going on for some people is like the Jackie. I like to call it the Jackie Chan neurons.
Did you say side quest?
Yeah.
The Jackie Chan neuron side quest. Okay.
Yeah. So call these the grandmother cells, but I like to call them the Jackie Chan ones because the first time that I saw them, it was this paper where they showed a photo of Jackie Chan. They did like the word like Jackie Chan, and then I don't know, maybe they said Jackie Chan, and this single neuron was like yes to all of those. But then when it gave like some other celebrity, it was like definitely not. Like even though it was a male person and like the sound was similar.
We know what he's been watching in his spare time.
So this was like, okay, this is amazing. And of course even though it is being encoded kind of like in a rate sense, like this single neuron is becoming more active over time, and it's not just like, totally quiescent and then just has one spike when Jackie Chan comes. It's like, it's just increasing its firing rate. But it's just so peculiar to me that it can be so eccentric, and clearly, well, I shouldn't say clearly, but it's in slight contradiction to this manifold hypothesis, which normally people that subscribe to this manifold idea, they sort of say, well, you can't get such detailed information from a single neuron, but you have to use a bunch of neurons. So it kind of relates actually to the superposition paper.
Yeah, right. I was even older than that. I think in the early days of neural networks, and even then, this deep learning up until the mid-2000s or something, the idea of distributed representations was taken as dogma. And I guess along the lines of what you just said, would have been viewed as not really paradigmatic for there to be individual neurons which stood for anything. But then as soon as people started training bigger networks, they very much noticed these things, and then people don't talk about distributed representations anymore. This term just disappeared, and now people just say representations as though that idea never existed. That's very strange somehow.
Yeah.
Yeah. So what is that just a similar, or how old is the evidence for these individual neurons which react to very specific inputs? Did that cause a bit of head scratching and reevaluation?
It's kind of still causes a bit of head scratching, but so from a biological perspective or like ecological perspective, it makes sense, I think, that you ought to have highly specialized things for stuff that matters, right? But for example, highly specialized neurons reacting to Jackie Chan, if Jackie Chan is important to you. But the classic kind of like hypothesis was that we should have these grandmother cells because, grandmother might be like a face that you see early in life and you're like that. But the sort of the problem with that from just from a more rational perspective is that, well, what happens if that cell gets damaged? Are you just going to forget your grandmother? Probably not, right? And neurons can easily get damaged. So, or, they can be plastic and change through, cause not really related to others. So, yeah. Yeah, I don't know. I don't know what to say exactly, but but the evidence is like the Jackie Chan neurons came out in 2020. They came out in that was discovered in 2020, but that was a pretty cool paper. So but it's pretty rare to get that sort of evidence in general because although we do sometimes put electrodes into people's brains, we don't do it for research purposes in the first instance. We do it for some sort of medical purpose and then sort of piggyback that with some experiments or some data recording. It's causing head scratching.
Is it really an individual neuron, or it means like probably one
one
neuron.
No, it's. I mean, from what I saw, I'll determine that was just one neuron. It could be other neurons. There could be a neuron for Jackie Chan's nose as well that's separate to the.
Can't we just
something
ask
like
this
that.
guy if we can pretty please knock out this one neuron and see what happens?
Just to test if, like, he remembers Jackie Chan afterwards, right?
Yeah.
Yeah. Yeah. So I don't know. Yeah, I really don't know. I'd be really. I think a lot of people would be quite surprised if that happened.
It seems impossible, right? So it seems like,
impossible. Yeah. Back up some latent storage somewhere else, but it's just it's quite surprising that a neuron would, like very specific, like just that it would be so specific, right? So high dimensional in another way of saying it.
You mentioned superposition a moment ago. Could you expand on that thought?
Yeah, well, so in general, and this is something I'm thinking about a bit more now and reading quite widely about, distributed representations and the manifold hypothesis in neuroscience and so on. And yeah, so I've been trying to think more about this. My thoughts aren't well formed, but the toy models of superposition seems to be just begging for a neuroscientific perspective on it, and I think, I mean, I don't know. There's a lot of different things to say, but what's kind of interesting is the more I've read from the neuroscience perspective, the more it's kind of like this natural science debate about which one is true in the case of the brain. And I'm not sure exactly what's useful to say.
Sorry, which one is true?
For the,
what do you mean? You're contrasting
like, a distributed
versus,
representation.
Yeah. Mm-hmm.
Yeah, more individual sort of coding or something like that. And also too, like I suppose the manifold hypothesis in neuroscience, like it carries this baggage around about rate coding and stuff like that. So I think actually probably a lot of people, including myself, would sort of be pluralistic about it and say, yeah, manifolds or rate coding obviously exists and is useful, but at the same time, these other coding schemes also work. And probably
regarding superposition, I mean, we understand a little bit beyond what's in that paper now, and it's sort of clear that there are energy costs to putting something in superposition and also to taking it out of superposition with a given set of pre-existing encoded features. So we can see phase transitions in some sense associated to both of those things, putting things into and taking them out of superposition. And you could kind of speculate that this is something that is actively managed. If it was, maybe it's happening in training in neural networks. Yeah, I would say almost certainly that's the case. But also in brains, perhaps, where you kind of maybe want to pack things. I mean, it's not an individual neuron, but like 2 or 3 or something, and you're putting things in superposition in that cluster. You might want to pack related things in the superposition, but then you change your mind and take them out and put them somewhere else or whatever.
Right.
So yeah, I'd be very interested if there's. I mean, what's interesting about the toy models example is sort of, I mean, the geometric nature of it is tied to the fact that there are these constraints, right? You're sort of trying to. It's basically like a packing problem, right? You're trying to put something in the structure, but it kind of has some tension so that it's not free to kind of shove it in there.
Right.
And if there's similar understood dynamics, I mean, I guess maybe there's something a little similar in associative memories, for example. I'm not sure, like where there's an energy cost to putting something into, like to represent it. And maybe, to come back to the distributed versus being in superposition. I mean, maybe if you have 2 modes of encoding something, and one is quite distributed and therefore relatively cheap, but maybe because it's distributed, low performance in some sense, there might be a trade-off where you like kind of take it out of a distributed mode and shove it into a more localized mode.
Right.
I don't know if there's any existing knowledge about that kind of thing in neuroscience. It'd be very interesting.
Yeah, I think that kind of is in like also not only just like learning and memory, but even developmental biology. I'm trying to think there probably is developmental neuroscience literature around this too, because, for example, it's quite famous that infants and young children have like a really huge number of synapses and connections, which they quickly like cull off and do like, I don't know, 90%, 99% pruning on in some cases. And kind of wondering, like if you treated that as a very extended sort of training process in an artificial neural network, or just considered that as an analogous thing that's happening for the child, then probably there are choices being made in different brain areas or in different situations that let's make this area a bit more of a distributed encoding area, or a little bit more of a superposition kind of situation, or a bit more localized, or whatever. It'll choose different coding schemes for different regions as appropriate, I think. Yeah.
Cool. I guess we should wrap up. Is there anything you want to say before we finish for this week?
No, I think that's good. I guess maybe just next week we can head more into AI land a bit, and if people have any other particular questions or other things, I can put it on Discord and I'll be happy to answer.
Yeah, sounds good.